Director of the Division of Clinical Innovation within the National Center for Advancing Translational Sciences within NIH. Prior to that, I was at the National Institute of Allergy and Infectious Disease working on vaccine, drug, and diagnostic development. Prior to that, a stint in industry doing drug development, and then past experience in academia doing clinical microbiology.
Thank you. Dr. Paul Offit.
Yeah, hi, good morning. I'm Paul Offit. I am a professor of pediatrics at Children's Hospital of Philadelphia and at the Perelman School of Medicine at the University of Pennsylvania. Thank you.
Great. Mr. Sheldon Tobin.
Good morning. My name is Sheldon Taubman. I'm an attorney at New Haven Legal in Connecticut, where I represent low-income individuals, mostly in the area of access to health care. But I'm here today in my personal capacity as a consumer representative for the...
Okay, thank you. Dr. Stephen Pergam.
Hi, I'm Steve Pergam. I'm an associate professor at University of Washington and Fred Hutchinson Cancer Research Center in Seattle, Washington, and I'm an infectious disease physician by training.
Thank you. Next slide, please. Mike. Mike, can you put the next slide, please? Thank you. Dr. Fuller.
Good morning. I'm Avita Fuller. I'm an associate professor at the University of Michigan in the medical school in microbiology and immunology and a member of the STEM initiative of the African Studies Center of the International Institute, and I'm a virologist by training.
Great. Dr. David Kim.
Good morning, David Kim. I'm the director of the Division of Vaccines in the Office of Infectious Disease and HIV/AIDS Policy in the Office of Assistant Secretary for Health, HHS. Thanks for having me.
Thank you. Dr. Eric Rubin.
Good morning. I'm Eric Rubin. Welcome from very snowy Boston. I'm a microbiologist at the Harvard T.H. Chan School of Public Health, an infectious disease physician at the Brigham and Women's Hospital, and editor-in-chief of the New England Journal of Medicine.
Excellent. Thank you. Dr. James Hildreth.
Good morning. I'm James Hildreth. I'm the president and chief executive officer of Meharry Medical College. I'm also a professor of internal medicine and a viral immunologist by training. Thank you.
Thank you. Next, Dr. Janet Lee.
Good morning. I'm Jeanette Lee. I'm a professor of biostatistics at the University of Arkansas for Medical Sciences, and happy to be here. Thank you.
Thank you. Dr. Mark Sawyer.
Good morning. I'm Mark Sawyer. I'm a professor of pediatrics at the University of California, San Diego, and Rady Children's Hospital, San Diego. I am a pediatric infectious disease specialist.
Thank you. Dr. Melinda Watson.
Good morning. I'm Melinda Horton. I'm director of the Immunization Services Division at the Centers for Disease Control, and I'm an adult infectious disease physician by training. Thank you.
Thank you. Dr. James Neaton.
Good morning. This is Jim Neaton. I'm a professor of biostatistics in the School of Public Health at the University of Minnesota.
Great. Dr. McInnes, Pamela McInnes.
Good morning. My name is Pamela McInnes. I'm retired as Deputy Director for the National Center for Advancing Translational Sciences, one of the NIH institutes.
Thank you. Dr. Patrick Moore.
Good morning. I'm Patrick Moore, Pat Moore, and I'm a professor at the University of Pittsburgh Cancer Institute and also in the Department of Microbiology and Molecular Genetics.
Thank you. Dr. Robert Shuley.
Good morning. I'm Robert Schooley, professor of medicine in the Division of Infectious Diseases at the University of California, San Diego.
Thank you. Dr. Stanley Perlman.
Good morning. I'm Stanley Perlman at the University of Iowa. I'm in pediatric infectious diseases and microbiology, and I have a long-standing interest in coronaviruses and immunology.
Great, thank you. Now I will do introductions for FDA staff. I would like to introduce Dr. Marianne Gruber, Director, Office of Vaccines, who will say a few welcome remarks. Dr. Gruber, please turn your camera and unmute your phone so everyone can see and hear you. Thank you, Dr. Gruber.
Yeah, good morning. My name is Marianne Gruber. I'm directing the Office of Vaccines Research and Review in the Center for Biologics Evaluation Research at the FDA. And I would like to welcome the committee members, Moderna, and the public to today's meeting. I want to thank the VRBPAC members for convening again today. We're looking forward to your thoughts and comments regarding the scientific evidence that will be presented by Moderna and the FDA. And we also look forward to your perspective on whether the benefits of Moderna's COVID-19 vaccine outweigh its risks to support authorization of the vaccine and an EUA for prevention of COVID-19 in individuals 18 years of age and older. So I look forward to the discussions and thank you.
Thank you, Dr. Gruber. I would also like to acknowledge the presence of Dr. Celia Witten, Deputy Director of CBER, and Dr. Philip Krause, Deputy Director of Office of Vaccines at this meeting, who may chime in as needed at a later point in the meeting. Also, Dr. Peter Marks, our Center Director, will join us shortly after I complete the reading of the conflicts of interest statement to make his remarks. Now I proceed with reading the conflict of interest statement. Thank you. The Food and Drug Administration is convening virtually today on December 17, 2020, the 163rd meeting of the Vaccines and Related Biological Products Advisory Committee, also known as VRBPAC, under the authority of the Federal Advisory Committee Act, FACA of 1972. Dr. Arnold Montoy is serving as the acting voting chair for today's meeting. Today, on December 17, 2020, the committee is meeting in open session to discuss the emergency use authorization, EUA, of the Moderna COVID-19 vaccine for the prevention of COVID-19 in individuals 18 years and older. The topic is determined to be a particular matter involving specific parties. With the exception of industry representative members, all standing and temporary voting members of the VRBPAC are appointed special government employees, FGEs, or regular government employees, RGEs, from other agencies and are subjected to federal conflicts of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflicts of interest laws, including but not limited to 18 United States Code Section 208, is being provided to participants in today's meeting and to the public. Related to the discussions today, all members, RGE and SGE consultants of this committee have been screened for potential financial conflicts of their own as well as those imputed to them, including those of their spouse or minor children, and for the purpose of 18 U.S. Code 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts and grants, cooperative research and development agreements, CRADAs, teaching, speaking, writing, patents and royalties, and their primary employment. These interests may include that are either current or under negotiation. FDA has determined that all members of this advisory committee, both regular and temporary members, are in compliance with federal ethics and conflicts of interest laws. Under 18 U.S. Section 208, Congress has authorized FDA to grant waivers to special government employees who have financial interests when it is determined that the agency's need for a special government employee services outweighs the potential for a conflict of interest. Waivers also may be authorized when the conflict of interest of a regular government employee is not so substantial and deemed not likely to affect the integrity of the services which the government may expect from the employee. Based on today's agenda and all financial interests reported by the committee members and consultants, there have been one conflict of interest waiver issued under 18 U.S. Code 208 in connection with this meeting. We have the following consultants serving as temporary voting members at this meeting today: Dr. Ovita Fuller, James Hildreth, David Kim, Janet Lee, Pamela McInnes, Patrick Moore, James Neaton, Stanley Perlman, Eric Rubin, Mark Foyer, Dr. Robert Shuley, and Melinda Watson. Among these consultants, Dr. James Hildreth, a special government employee, has been issued a waiver for his participation today to participate at the meeting. The waiver was posted on the FDA website for public disclosure. Dr. Greg Sylvester of Securus Incorporation will serve as the alternate industry representative for today's meeting. Industry representatives are not appointed as special government employees and serve only as non-voting members of the committee. Industry representative of this committee is not screened for financial conflicts of interest and do not have voting privileges. Also, industry representatives act on behalf of all the regulated industry and bring general industry perspective to the committee. Mr. Sheldon Taubman is serving as the consumer representative for this committee. Consumer representatives are appointed special government employees and therefore are screened and cleared prior to their participation in the meeting. They are voting members of the committee. Today's meeting has one external speaker, Dr. Stephen Goodman, who will serve as the guest speaker. He has been asked to disclose any financial interest he may have related to the product before the committee. Disclosure of conflicts of interest for guest speakers follows applicable federal laws, regulations, and FDA guidance. FDA encourages all meeting participants, including open public hearing speakers, to advise the committee of any financial relationship that they may have with any affected firm, its products, and if known, its direct competitors. We would like to remind standing and temporary members that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to inform the DFO and exclude themselves from such discussions, and their exclusion will be noted for the record. This concludes my reading of the conflict of interest statement for the public record. At this time, I would like to invite our Center Director, Dr. Peter Marks, to make a few remarks welcoming the committee. Dr. Marks, please could you turn your camera on and the speaker, unmute your speaker phone, and the floor is yours now. Thank you. Go ahead, Dr.
Good morning. Thanks, Prabha. Good morning. I'd like to take a moment, first of all, to welcome you all and also to provide a brief overview of advisory committees and the role they play in assuring transparency in FDA's decision-making processes. FDA uses advisory committees to obtain advice from experts who work outside of the government. It does so while working towards an open and transparent government by presenting information under consideration in a public forum and encouraging patients, healthcare providers, and other interested people to share their views during the open public hearing or by submitting comments to the docket. A key part of FDA's mission is to evaluate new therapies and determine which are safe and effective for their intended uses. This complex job often involves many areas of expertise, and sometimes FDA turns to outside experts for counsel, such as for the COVID-19 vaccine under consideration today. Advisory committees weigh the available evidence and provide scientific and medical advice to the FDA on the safety, effectiveness, and appropriate use of products that the agency regulates. FDA advisory committees are just that, advisory in nature. It's important to note that the advice that the FDA receives from the committee does not represent the position of the FDA. Rather, the FDA weighs the advice that it receives when taking actions on medical products. FDA ultimately makes the final decisions on all matters that come before the committee. Also, to set expectations, for today's meeting, we've organized the agenda topics slightly differently than last week's meeting to allow the committee members to have sufficient time for a robust discussion of the questions before them. We invite the public and the committee to review the presentations and recording of the December 10th meeting for more information on COVID-19 epidemiology, vaccine safety and effectiveness monitoring, and operational distribution plans, as those will not be covered in depth today as they were at the last meeting. So as we begin today's proceedings, I want to take the opportunity to thank you all, including all of the advisory committee members, for the insights that they'll provide, and also thank the FDA staff, the sponsor, and those presenting at the open public hearing today for participating. Your contributions are very important in helping us at well-reasoned, science-based decisions. Thanks very much, and we look forward to the meeting today.
Okay, great. Thank you, Dr. Marks. Now I would like to hand over the meeting back to our chair, Dr. Ana Armanto. Dr. Armanto, take it away.
Thank you very much, Prabha. First, we're going to hear from Dr. Doron Fink, Deputy Director of the Division of Vaccines and Related Products Applications at FDA, who will give us a presentation on emergency use authorization. Dr. Fink.
Hi, good morning. If the AC staff could please make me a presenter, then I will begin my presentation. In the meantime, I'll introduce myself. I'm Doron Fink. I'm the Deputy Director for Clinical Review in the Division of Vaccines and Related Products Applications, Office of Vaccines Research and Review, Center for Biologics Evaluation and Research at FDA. The COVID-19 pandemic continues to worsen in the U.S. and worldwide. As of the week ending December 15th, there have been a total of 16 million cases and greater than 300,000 deaths in the U.S. to date, and 1.5 million cases and greater than 17,000 deaths just in the past week. On December 11th, just last week, FDA issued an emergency use authorization for the Pfizer-BioNTech COVID-19 vaccine. This vaccine is authorized for active immunization for prevention of COVID-19 due to SARS-CoV-2 in individuals 16 years of age and older. The EUA was issued after a December 10th VRBPAC meeting to discuss the vaccine, data informing its benefits and risks, and plans for its further evaluation. On November 30th, Moderna Therapeutics submitted an EUA request for the Moderna COVID-19 vaccine, otherwise known as mRNA 1273. This, like the Pfizer vaccine, is an mRNA lipid nanoparticle vaccine, and it is administered as a 2-dose regimen 28 days apart. The requested use for this EUA is for active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 18 years of age and older. The information submitted with the request includes safety and efficacy data from a large, randomized, blinded, placebo-controlled phase 3 trial. FDA has been conducting a comprehensive review of the Moderna COVID-19 vaccine EUA submission received on November 30th. As with the Pfizer request, our review has been comprehensive and conducted over a short period of time. We have verified clinical data integrity and integrity of Moderna's analyses and conducted our own independent analyses from data sets provided in the submission. We've conducted ongoing review of manufacturing, non-clinical, and clinical assay information. including information that has come in the last few days. We have reviewed and worked on revisions of prescribing information and fact sheets necessary to inform vaccine recipients and healthcare providers. We have had multiple information requests to Moderna to address our questions and need for clarifications. And of course, we have prepared for today's VRBPAC meeting. This will sound like a bit of a broken record, but I say it again because it's important. Today's VRBPAC meeting continues FDA's commitment to an expedited review process that is transparent, scientifically sound, and data-driven. As a reminder from material presented last week, the legal authority for emergency use authorization was established in Section 564 of the Federal Food, Drug, and Cosmetic Act and allows for FDA authorization of unapproved medical products or unapproved uses of approved medical products to address public health emergencies related to biological, chemical, radiological, or nuclear agents. HHS Secretary Azar issued a declaration on March 27th justifying emergency use authorization of drugs and biological products to address the COVID-19 pandemic, which is a necessary prerequisite for issuance of an EUA. Here again are the criteria for FDA issuance of an EUA. The agent referred to in the EUA declaration must cause a serious or life-threatening disease or condition. Again, we know this to be true for COVID-19. The medical product must be effective or must be believed to be effective to prevent, diagnose, or treat the serious or life-threatening disease or condition caused by the agent. The known and potential benefits of the product should outweigh the known and potential risks of the product. And also there must not be any adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. As I explained last week, there is only one FDA-approved product for COVID-19, which is remdesivir, approved for treatment and not for prevention. As I mentioned at the beginning of my talk, the Pfizer-BioNTech COVID-19 vaccine is now available under EUA for prevention of COVID-19. But it remains unapproved, and its quantity is not sufficient for mass vaccination needed to address the pandemic in the U.S. Therefore, the fourth criterion is still met. FDA explained in guidance and in the VRBPAC meeting on October 22nd our expectations for data and other information to support issuance of an emergency use authorization for a COVID-19 vaccine. This information includes data to demonstrate manufacturing quality and consistency. And similar to the case with the Pfizer vaccine last week, FDA has reviewed the manufacturing information provided by Moderna and found it to be adequate to support issuance of an EUA. We expect clear and compelling safety and efficacy data to support a favorable benefit risk of the vaccine when rapidly deployed for administration to millions of individuals, including healthy people. And finally, we expect plans for further evaluation of vaccine safety and effectiveness, including in ongoing clinical trials, active and passive safety monitoring during use under EUA, and observational studies. Last week I had a number of slides outlining more details of these expectations. In the interest of time, I'm going to skip those today. If an EUA were to be issued for the Moderna COVID-19 vaccine, it would specify the conditions of use for which benefit risk has been determined to be favorable based on review of the available data. These conditions include the populations to be included in the EUA, conditions for vaccine distribution and administration, and requirements for safety monitoring and reporting of adverse events. Vaccine made available under EUA will also be accompanied by information for vaccine recipients and healthcare providers by way of prescribing information and fact sheets. These will describe that the vaccine remains unapproved and under investigation under IND, They will describe the known and potential benefits and risks of the vaccine, and will also discuss available alternatives and the option to refuse vaccination. As I explained last week, an EUA that is issued may be revised or revoked for a number of reasons. If circumstances justifying the EUA no longer exist, if criteria for issuance are no longer met, or if other circumstances arise that warrant changes necessary to protect public health or safety. For example, based on new information concerning vaccine safety or effectiveness, vaccine manufacturing or quality, or COVID-19 epidemiology or pathogenesis. I want to pause here to address the issue of anaphylactic reactions or serious allergic reactions following vaccination. While today's discussion is about the Moderna vaccine, at last week's meeting we reported on anaphylactic reactions that occurred in the United Kingdom in 2 recipients of the Pfizer vaccine, which is also an mRNA and lipid nanoparticle vaccine and therefore relevant to today's discussion. Both of these vaccine recipients had a medical history of serious allergic reactions, though not, as far as we know, to any of the vaccine components. Yesterday, as has been reported in the press, 2 healthcare workers in Alaska experienced allergic reactions minutes after receiving the Pfizer vaccine. One of them an anaphylactic reaction resulting in hospitalization. All of these individuals were treated with appropriate medical interventions, and thankfully, all are recovered or recovering. We anticipate that there may be additional reports which we will rapidly investigate. We learned of these cases through established safety surveillance systems that worked exactly as designed. And FDA is coordinating with CDC to further investigate the cases in the U.S. and to communicate our findings in a timely manner with vaccine providers and recipients. FDA and CDC are also in close contact with public health and regulatory authorities in the United Kingdom as they continue their investigations. While the totality of data at this time continue to support vaccinations under the Pfizer EUA without new restrictions, these cases underscore the need to remain vigilant during the early phase of the vaccination campaign. To this end, FDA is working with Pfizer to further revise the fact sheets and prescribing information for their vaccine to draw attention to CDC guidelines for post-vaccination monitoring and management of immediate allergic reactions. This revision will be in addition to the information already included in the contraindications and warnings, including that facilities where vaccines are being administered should ensure that medical treatment for managing serious allergic reactions is immediately available. We will do the same for the Moderna vaccine should it be authorized for use under EUA. Here's the agenda for today's VRPAC. As Dr. Marks mentioned, we have a lighter schedule than last week to allow for more robust discussion. You'll see that some of the presentations from last week are absent because the information is not materially changed. We will have a repeat of Stephen Goodman's talk on considerations for placebo-controlled trial design if an unlicensed vaccine becomes available. I'll explain the reasons why on my next slide. Following Dr. Goodman's talk, we will hear a sponsor presentation of the data for the Moderna COVID-19 vaccine. We will then have an open public hearing followed by a lunch break. And finally, an FDA presentation of our EUA review, discussion items, and questions for the committee to discuss and vote. We have just one question today for discussion without a vote. This question is similar to one we asked last week, but we've rephrased it in a way that we hope will focus the discussion. The reason we are coming back to this question is because it's important. A case-driven vaccine trial conducted in the midst of a pandemic that very quickly demonstrates clear evidence of efficacy, at least in the short term, can allow the vaccine to be made available under EUA. On one hand, this has the very positive effect of helping to address the pandemic. On the other hand, widespread vaccine availability can interfere with conducting the trial to completion. To be clear, FDA has never insisted that placebo recipients enrolled in ongoing trials who want vaccine be made to wait beyond when the vaccine would otherwise be available to them under the conditions of an EUA, prioritization recommendations, and available supply. Rather, we have been asking those responsible for conducting COVID-19 vaccine trials to think carefully and creatively about how to continue trials after vaccine becomes available under an EUA, to preserve whatever societal value can be preserved, and to ensure that sufficient data are ultimately accrued to support vaccine licensure. This includes encouraging study participants who are willing to remain in blinded follow-up for the same altruistic reasons that prompted their enrollment in the first place to do so. Later today, you will hear about Moderna's plans for their trial. The question that we would like you to discuss is, in considering Moderna's plans for unblinding and crossover of placebo recipients, please discuss the most critical data to further inform vaccine safety and effectiveness to support licensure that should be included in either ongoing clinical trials with the Moderna COVID-19 vaccine or other studies such as additional clinical trials or observational studies with that vaccine. Following this discussion, which again will not have any vote, we will have a single question for VRBPAC discussion and vote. And that question is, based on the totality of scientific evidence available, do the benefits of the Moderna COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older? Thank you very much.
A full 20. Thank you, Dr. Fink. We have a full 20 minutes for discussion here. I think we should restrict our discussion to the EUA process and its characteristics. Since we're going to be hearing from Dr. Goodman about some of the other issues, we probably should restrict questions or discussion about that until after he presents. So raise your hands, please, if you would like to make a comment. And Dr. Meisner.
Thank you, Dr. Monto. First, I would like to express my gratitude to Dr. Fink, Dr. Marks, Dr. Gruber, and their colleagues at the FDA for the extraordinary amount of work that has been put into this issue over the last few weeks and months. I think that all the citizens of the United States should recognize the enormous effort that has been put into this. So thank you. My question is as follows, and it's somewhat similar to the question that was asked, that I asked last week. It's important that we move a vaccine from an EUA to a BLA because there are a number of advantages to have a vaccine licensed and recommended by the CDC. Is there any way you can anticipate how soon that might happen? And does the availability of a second messenger RNA, a vaccine with a similar mechanism of action, will that facilitate the decision in any way for the FDA? Over.
Thank you for that question. As I believe I responded last week, we are actively working with the vaccine manufacturers, both Pfizer and Moderna, to arrive at a data package that would support vaccine licensure. This data package would include some additional follow-up from clinical trials, as well as data accrued from use under the EUA, as well as some additional manufacturing information for vaccine that is intended to be produced following licensure. So it is our goal to arrive at a licensure application as quickly as possible as the data allow. And in terms of your other question, certainly two vaccines that are similar in platform, although not exactly the same, will be considered relevant to each other and will inform our assessment of those respective vaccines.
Thank you.
Dr. Carrillo.
Thank you. Doran, I want to make sure that I understood what you said. You seem to imply that the issuance of a second EUA was partly dependent on the fact that there was inadequate supply of the initial EUA for mass vaccination. Is that a criteria that would potentially impact a decision on future EUAs for other vaccines?
So thanks for the opportunity to clarify that question. So actually, the supply of Pfizer vaccine is secondary at this time for considering issuance of an EUA for a different vaccine, and that's because the Pfizer vaccine is not approved. So consideration of available alternatives requires that those alternatives both be approved and adequate. So the fact that the Pfizer vaccine is not approved means that there is currently no approved, available, and adequate preventive vaccine for COVID-19.
Thank you.
Dr. Rubin.
Thanks, Dr. Fink, for that very clear presentation. I'm curious what FDA will do with the discussion item on Dr. Goodman's proposal. Is it likely to end up as an FDA requirement or a strong recommendation to proceed to BLA for the manufacturers?
Well, as I explained before, we are working actively with the vaccine manufacturers on accruing data that would be necessary to support a biologics license application. And this includes discussions around the contours of their ongoing clinical trials going forward. We are hoping that the committee discussion will help to inform those discussions with the manufacturers.
Dr. Perlman.
Yes, so I just have a question about one of the last things you were talking about. So the anaphylactic reactions have clearly been a big deal in the press, and I and probably others get lots of calls about what it means. And I think the FDA recommendations talk about allergies to components of the vaccine, yet the components of the vaccine actually are not obviously, to me, allergenic. So do you have any sense for how the FDA is going to finally make recommendations? The UK has different recommendations than the FDA came out with. So do you know how that's going to play out, and do you know what the components are in the vaccine that could be inducing this?
So at this point, we and CDC are continuing to investigate these cases and consider data. At this point, we don't have enough information to make definitive recommendations one way or another. And as we continue to investigate and evaluate the data, we will consider whether additional recommendations need to be made.
Dr. Pergam.
Thanks, Dr. Finks, for the clarity again and for a short presentation, because I know we have a lot to discuss today. I had a question. You brought up the issues of a couple of separate questions in addition to the main EUA question that we're going to be reviewing related to what other studies need to be done, et cetera. I I want to be clear, is this for both vaccines, since we did not get to review those and give those recommendations to the Pfizer-BioNTech vaccine? So those discussions for additional studies or additional work that needs to be done, are those going to be relevant for both vaccine candidates?
Well, they certainly will be relevant for both vaccines. We'd like this discussion today to focus specifically on Moderna's plans. But clearly, the ideas discussed will be relevant to both vaccines.
Thank you.
Dr. Moore.
You know, for the long-term safety, meaning beyond years, even decades-long safety for this vaccine and the other vaccines, requires obtaining a centralized resource that allows us to know who was vaccinated and who was not. Is that being planned to be collected for outside of the randomized controlled trial? Is there a plan to collect that information to securely store it so that you can do linkage analysis with, say, cancer registries or autoimmune registries?
So as discussed last week, the U.S. government is planning a number of studies leveraging healthcare claims databases to evaluate vaccine safety over the longer term with use under an EUA. I'm not the expert on those studies, and so I would have to defer comment on the details to those who are spearheading them.
Dr. Fuller.
Thank you, Dr. Amato, and thank you, Dr. Fink, for your explanations. The question, which may be addressed later, but I'll ask you, how will the FDA or CDC or other state health agencies monitor the potential adverse events that happen, like the allergies that you mentioned in Alaska? If it were a continuing clinical study, those would be picked up by the researchers. But in this case, how will that be done? Could you please share a little bit more?
Sure, and this was also explained last week in one of the presentations by CDC that we don't have today, but I'll refer you back to and refer the public back to the recording of that presentation. We have robust safety, surveillance, and reporting systems that have been in place for a long time, including VAERS, the Vaccine Adverse Event Reporting System. Additionally, vaccine recipients under the EUA will be asked to partake in a program called vSafe, which is an electronic safety reporting system that the government is using to track vaccine safety with use under the EUA.
All right, thank you.
Dr. Neaton.
Thanks, Dr. Fink. My question is actually similar to Dr. Pergam's. Have you considered aligning some of the future protocols for these two vaccines in a manner, and also the current protocols with the purpose of being able to combine the data from the studies, two pivotal studies?
Well, yeah, combining or pooling data involves complicated statistical considerations, but what we have done, and we discussed this at our October 22nd VRPAC meeting and also in our guidance released in June of this year, is we have recommended standardized case definitions that will help to evaluate efficacy results from trials of different vaccines. Not necessarily for comparing one vaccine to another, although that is one possibility. We hope that this standardized approach, which as we explained in October is not a requirement for the primary endpoint, but a recommendation that we've made for inclusion in all of the phase 3 trials. We hope that this will facilitate the type of broad and robust data analysis that you might be thinking of.
Yeah.
Dr. Gantz.
Thank you very much. Thank you, Dr. Fink. I had one question about, I realized today we're entertaining the Moderna vaccine. We've now entertained the Pfizer one previously. And I'm wondering, in the context of other vaccines that are coming to the market, all of which are going to have different adverse events as well as different populations in which they should be used, I'm wondering in the context of equity in terms of how we roll these out. Some are coming to obviously into use before others. And I just worry a little bit about how we should think about that in the context of the broader field of different vaccines that are coming that have different profiles. If you had any thoughts on that, or how the FDA and CDC are thinking about those other vaccine models in the context of this.
Sure. So just to be clear, FDA's responsibility is to evaluate data concerning the benefits and risks of the vaccine in the context of the emergency use authorization request. And once FDA issues an emergency use authorization, then the responsibility falls to CDC and the Advisory Committee on Immunization Practices to set prioritization recommendations and other recommendations for use of the vaccine, considering its benefits and risks and the populations included in the emergency use authorization.
Dr. Chatterjee.
Good morning. I have a question with regard to the BLA applications that may be coming from the manufacturers. In the past, from my experience anyway, usually a BLA application comes in after the clinical trials have been completed. For these vaccines, is there a plan? I couldn't quite understand from your explanation, Dr. Fink, whether there will be interim data analyses and we won't actually have to wait until the trials are completed before the BLA applications will be entertained by the FDA.
Yes, thank you for that question and the opportunity to provide clarification. So it's actually not unusual for a clinical trial to be ongoing for longer-term safety and or effectiveness follow-up when a biologics license application is submitted. At this point, we do have interim data for 2 COVID-19 vaccines, one of which we have authorized for emergency use, another of which we are considering today. At this point, the data would not be considered quite sufficient to support a biologics license application, but as I mentioned before, we are working with both manufacturers to accrue the data that would be needed, with the goal of getting these vaccines licensed as soon as the data allow. We heard from Pfizer last week that they are anticipating potentially submitting a biologics license application sometime in the spring of next year, and that plan is certainly within the realm of what we would consider possible.
Thank you very much.
Dr. McInnes.
Morning, Dr. Fink. I have a question regarding the status of inspection of facilities. And the reason I think this becomes important for us to have some sense of where you all are in this was this availability of information on the news about unexpected volumes left in syringes. So while that may very well just be due to residual volume in syringe and needle of particular type versus what might be seen in another situation, it does bring up the question of the confidence in the manufacturing sites, the manufacturer, the fill, the consistency. And while I appreciate that a full picture of that is not required under EUA, I would like to have some sense of the sort of confidence of the FDA in that particular state of data.
So I can repeat that we have reviewed extensive manufacturing information for this EUA request and do feel confident that we have enough information to justify issuing an EUA for this vaccine should everyone agree that the benefits outweigh the risks based on the clinical data. I can't speak in more detail about facilities inspections. I'd invite other FDA colleagues who might be on the line to chime in if they have something additional to say. The other thing I'll mention about the volume issue for the Pfizer vaccine is that if you look at the instructions, they are to add 1.8 mLs of diluent to 0.45 mLs of vaccine that's already in the multi-dose vial. That gets you to a total of 2.25 mLs. And so with a dose volume of 0.3 mLs, it is actually not at all unexpected that there would be more than 5 doses in those vaccines, or in those vials. Over.
Any further FDA comments? Okay, let's, for the final question, Mr. Taubman.
Thank you for the excellent presentation, Dr. Fink. I have a question about the FDA's review of efficacy data. Your slide indicated a November 30th submission for EUA. But the briefing documents indicate that Moderna submitted another set of documents on 12/7, and that included, so instead of a closed date of 11/7, it's a closed date of 11/21. And in the briefing document, you indicated FDA had reviewed some and verified some of that more recent data, but not all. Could you just briefly explain what that actually means? And there's been reports in the press about how the FDA's process is more rigorous than the British review process, for example. But just briefly explain what that really means. And second, to confirm that you have been able to verify the efficacy data in the second set regarding the primary endpoint as well as the important secondary endpoint of preventing severe disease.
Yes, we'll hear more about that in our FDA presentation this afternoon, but I can verify, I can confirm that FDA has examined, verified the integrity of, and confirmed the efficacy analyses from the later time point, from the November 21st time point, both for the primary efficacy analysis and for the secondary analysis of severe cases. What we have not done due to time constraints is more in-depth probing of the data to do our own independent analyses on some questions that we look at that are maybe not so central but of interest. So all this to say that we have verified and confirmed Moderna's analyses for both the interim and final efficacy analyses, those that we consider to be most critical to inform the benefit-risk assessment. We have not done quite as comprehensive a dive into those data as we did for the interim analysis, but we don't think that this should hinder in any way our confidence in the data to support an assessment of benefit and risk.
Thank you. Okay, we're moving on now to the presentation from Dr. Stephen Goodman. And he'll be telling us about the considerations for placebo-controlled trial design if an unlicensed vaccine becomes available, Dr. Goodman is Associate Dean of Clinical and Translational Research at Stanford University School of Medicine. Dr. Goodman.
Morning. Can you hear me fine?
Yes.
Okay, terrific. So thank you so much for inviting me, and in fact, particularly for inviting me back again. I think the reason for that was presaged by the questions and talk just given, which is that the issues we'll be considering here are not just relevant for this vaccine, but for many trials currently ongoing and those in the future. So this arguably has potentially more long-lasting and effect than even the EUA today. I don't want to scare you with this title, which looks exactly the same as last week. I'm in fact not going to be giving exactly the same talk. I'll be picking up from where we left off last week and taking a bit of a deeper dive to give you more material for your discussion. And this is the outline. I'll very briefly remind you where we left off after last week, then go into the Moderna consent and proposal. Then we'll take a bit of a deeper dive into the deferred vaccination design, which we talked about last week. Then I'll discuss the evidential and ethical effects of both partial and complete unblinding of the placebo group, which are both under consideration right now. And then I'll have a final single slide on the evolution of design, which is a perspective I hope you will consider in your comments. And I already heard a suggestion of this from Dr. Gans in her last question. So let's go just to very briefly summarize where we were after last week. We had an ethical summary where we talked about the issue of ethical dilemmas as being a choice between two right actions or justified in different ways, which is certainly what we're going to be facing today, even more starkly. The importance of trust in the whole vaccine development process and prioritization, which enables us to do these clinical trials and could be withdrawn at any time. Talked a bit about the issue of context, where the ethical calculus depends not only on what we know and what we don't, but the availability of vaccine. And we talked about ethically relevant benefit in the sense that whether the placebo group was taking on such a risk relative to the vaccinated group that they were owed something just by nature of that benefit. And the argument was that they were not, even though there was some very, very small deficit. But of course, you don't know that when you sign up for the trial. We only know that in retrospect. In terms of the epistemic or evidential summary, I'm going to use the word evidential more today than epistemic, even though I'm really talking about the same thing, which is that all designs can generate valid evidence, albeit with different efficiency and degrees of certainty. That's randomized controlled trials, quasi-experimental and observational designs, and in particular knowledge of mechanism and biology, which guides a lot of the interpretation of the empirical design. And finally, that RCTs are best to assess some properties, vaccine properties, but not necessarily all. So they're very good for some things, but we have to partition between the things it's good for and not. And finally, the idea that there shouldn't be any bright lines drawn either on the ethics or the epistemology fronts, and we shouldn't be declaring anything particularly unethical. What we're really saying when we use that word is we believe that one principle outweighs another, and the word unethical sort of disenfranchises people on the other side and demonizes them, and doesn't lead to good discussions, as well as strict adherence to randomization when that's not necessary. So let's go into the Moderna consent and proposal. The consent is at the beginning exactly what you would expect. It mentions voluntariness, that particularly important that the participant may or may not benefit from participating in the study, but it is designed to help others in the future, that they can leave at any time. This won't affect their future care. Then we get into some of the questions which state in very plain language what the participants should expect. And it says what will happen at the end of the study. Basically, you'll just be discharged from the study by your doctor. Will you be informed if new information becomes available? Yes, and certainly, as with last week, the EUA is part of that. But this is the most important clause here at the bottom. Can you continue getting the study vaccine after the study? And this is what was told in the consent. If you choose to withdraw from the study or are taken out of the study, you will not continue receiving the study vaccine. Also, if the study is terminated early or when the study is ended, sponsor will not continue providing the study vaccine. Now that will be highly relevant to the actual proposal, which we will contrast here. So first of all, an observation is made that there's going to be a large number of folks who are eligible for early vaccine administration. This, in particular, 25% of the enrollees are healthcare workers. But here's the proposal: they will the vaccination, and then they will be observed unblinded for the rest of the entire 2 years. And adverse posing to simply unblind and immunize the placebo group. This is importantly different from last week's proposal, which was to wait until there was until they were eligible for receipt otherwise, and then if they asked, they would then be unblinded and immunized, and that all participants would be encouraged to stay in the randomized trial as long as possible, and that everybody would be immunized at the end of 6 months. So this is different in multiple ways. First, that it's done immediately. Second of all, that they be unblinded, that actually is common to both, but that we don't wait for eligibility outside the trial. So this is a very, very, very important difference, and something that will have, as I will argue, consequences not just for this trial, but for other trials of other vaccines. So this is exactly the same as what we talked about before. What is owed to placebo participants, and this really is what is the obligation, and that's something that can be asked of the investigators or the company because it's owed to them. And the simple answer is what's in the consent. That's what's owed to the placebo participants, the conditions on which they enrolled, and of course all the adherence to the Belmont ethical principles. They certainly would want to expect that they wouldn't be denied vaccine if it became available to them, which could be done through an exclusion, the EUA, but I don't think that is actively being considered. And potentially, reciprocity, which is really a form of gratitude and not obligation, could be operationalized through higher priority for vaccine within their priority group when they become eligible. But there's not an ethical obligation of investigators to unblind on demand. It is if there's a medical reason, but not for others. And not immediate vaccination in the trial before their turn is called outside the trial. And that's actually reflected in the consent. If this was an ethical obligation to immediately vaccinate once there was interim results showing efficacy, or even with an EUA, this would have been in the consent, and it wasn't. So let's now take a little bit of a deeper dive into the vaccination, into the deferred vaccination design. I showed this slide last week. There are really two alternative designs at this point that are that might be considered for this or future trials. One is this deferred immunization, which is a blinded crossover design, and the second is active control designs. And I'm not going to focus on those, but I will talk about the implications of the guidance on this placebo group for the future of active control designs. That's very important. And of course, everything comes with active and passive observational studies of almost every aspect of the vaccine, not just safety, the vaccine properties. So both approaches are going to require some give on the evidential and ethical side. So this is a picture you saw last week. This is the deferred vaccination arm, and on the top you see the
the arm that gets immediate vaccine, that's in blue, that narrows, and the narrowing reflects a slow waning of the vaccine efficacy. Of course, we don't know if or when that happens, but this is just a schematic to show you how they can be still compared if we cross over blindly. On the bottom, you see the placebo arm, and the point at which there's early efficacy established, which is roughly where we are today, is still in period 1. And then the proposal is that at some point, if somebody is going to become eligible for vaccine, that if they're in the vaccine arm, that they get a placebo injection, and that if they're in the placebo arm, they get a placebo. I'll get this straight. If they're in the placebo arm, they will get a vaccine injection. So everybody ultimately gets immunized, but they still don't know whether they were in the immediate vaccination arm or in the deferred vaccination arm. And this preservation of blinding, even with the immunization of everybody in the trial, allows a number of things that I'll talk about in a minute, and I mentioned last week. So this is another way to represent what's going on. And here we're measuring efficacy not as the thickness of a bar, but in terms of attack rate. And the attack rate is on the vertical axis. And what you see is the attack rate you'd expect under placebo in the early days, and on the bottom is just time, would be high. Here it's just nominally indexed at 1. That would be the attack rate in the red line of the placebo group, and at the bottom would be the attack rate in the vaccine group. In fact, in practice, it's turned out to be much, much lower, roughly 95% lower than the red line. And then we watch them over time, and the vaccinated arm, if vaccine efficacy starts to wane, if and when, what you would see is a slow rise in the attack rate in the vaccinated group, and that blue line starting to ramp up. But of course, there are other things going on in the world that affect the attack rate, including prevalence in the community, community restriction measures. So there's a lot of things going on in calendar time that need to be controlled for, which is why we can't just observe what happens over time. We won't know if the attack rate starts to go in the vaccine arm exactly what it's due to. So this is where the deferred vaccination can still help us recover that information. And you see that vaccination occur with the big drop at the point of deferred vaccination from the red line down to a new blue line. And we watch this attack rate for a while. And if there's a difference between the attack rate in the early vaccinated group versus the late, that is sign that there's waning of efficacy. And of course, this can occur at every point. They could be at the same level for a while, and then the early vaccinees start to, their efficacy starts to wane and their attack rate goes up over time. It doesn't have to be right at the point of crossover. So this is what it looks like, and this is how you can recover this really critical information about how long this immunity lasts, which is going to be a major question, and that we will also see that there are a few other things we can do with this design. So I want to make the point that the crossover can occur whenever an individual participant becomes eligible for an available vaccine outside the trial. So it's not occurring for everybody at the same time, which those schematics suggested. It's occurring for each individual potentially at a different time. I'll talk more about that issue of design. There's also some unexpected benefits. One is that blinded crossover allows for more safety assessment via self-control design. And you'll see here on the bottom that for the placebo arm, we're looking very, very carefully at adverse event incidence in the post-placebo period. That serves as a control for the AE seen in the vaccine arm, which I've sort of erased up here. I'm only looking at the placebo arm. You'll see why. And then when they cross over blindly, of course, they don't know that they're crossing over. We can then watch very closely adverse event incidence post-vaccine, and we can compare the AEs after this vaccination to before in a particularly
powerful way, which is with control within each individual. This is not just comparing overall incidence rate, which is like what's occurring in that first period. It's a self-controlled design, which controls for confounding in a particularly powerful way. So this is a nice benefit of this design because they're being watched carefully in that first period. So this is very solid AE information. We could ask people if they had a recent stroke or heart attack or whatever before entering the trial, but for certain AEs might not remember it very well, and certain ones might prevent somebody from enrolling. So this guarantees that there's absolutely no bias about who's included. There's another bonus, which is that as vaccine efficacy wanes, if it does wane, the deferred vaccination allows a booster trial sort of right on top of the trial infrastructure, and is, in that sense, is an added benefit if we find that a second booster, that is a third shot, is needed, and this can be piggybacked right on top of the deferred vaccination structure. So this again is a very nice add-on if needed. Now, last week that company was asked about whether they were amenable to doing this. They pointed to the logistics of maintaining the blind. They said it was very difficult and maybe not worth it. I will say that there are additional logistics, and it will be up to you to explore the yield and to give FDA advice on whether this is determinate and whether it's worth instituting. So there's mandatory crossover serology plus a dummy shot, and then there's another dummy shot for both arms, because neither one can know which arm they were in. There are possibly more blood draws, and these have to be synchronized between the two groups. They should be kept on the same schedule, so serology is comparable. And finally, there is re-consent, but this is necessary for any major design change, including unblinding and administration of vaccine. So this is not necessarily an additional logistical barrier. But what's really critical is that in theory, no one knows their assignment. And all the parameters, all the reasons we do the RCT apply here in preventing bias and in ascertainment of a whole bunch of AEs, efficacy endpoints, and even trial participation, as I will mention. So this is a complicated slide. I'm actually not going to go through it in detail. I'm just going to point out two things. This shows us, this is a list of all the things we still want to learn about the vaccine that is not really captured very well at this interim point, which includes duration of immunity after 2 months. This actually, under certain circumstances, can be enhanced by the deferred vaccination design, even over continued placebo control, even though I think that's off the table. And the other thing that's enhanced, which is really critical, and this is the link to the future, is correlates of immunity, because we're now doubling the vaccinated arm. We're doing so in this randomized way, and we can enhance the finding of markers, surrogate markers of protection, which will be absolutely critical for active control designs in the future. I've mentioned a variety of things here that are partly preserved. That is, we'll be able to get some information, but not necessarily definitive information. Now, finally, effects of complete or partial placebo unblinding, both on the evidential and ethical scales. In terms of partial unblinding, which would be done if we gave the vaccine to those who, at the point they became eligible, so we would never completely unblind the group until maybe some time point in the future, as Pfizer recommended, but not as Moderna is proposing. So it's important to mention the same fraction of vaccine recipients would also be unblinded, because when they ask to be unblinded or when they become eligible, they don't know what group they're in. So both people in either group will be unblinded. We lose them both, at least to the randomization. The remaining cohort will probably be at lower risk, as the higher risk ones will be the ones we will either potentially
they will request unblinding, or we will offer the unblinding because they become eligible. Once a vaccinated person realizes they've been vaccinated, they will probably engage in higher risk behavior, which will, for at least the 1 to 2 months after the unblinding, will make them a more difficult comparison group to the placebo group. And finally, patient-reported outcomes for the unblinded group can be biased in terms of how people interpret various symptoms. There'll be an impaired ability to evaluate waning vaccine efficacy in the first 6 to 8 weeks after crossover. After 8 weeks, everybody knows they've been vaccinated, but before this time, that vaccine efficacy is uncertain. And it has unpredictable effects on trial retention, particularly safety assessments to the crossover, because once you do the unblinding, there's a sense that, well, the experiment is over. You know, you're coming back, giving information, but we don't have the same sort of bonding to the trial in the sense that adherence to its to the dictates are actually critical for validity. If we're going to completely unblind the placebo group, then we lose a lot from the evidential side. There's no comparison group to compare rates of infection or safety, so the duration of protection, long-term hazard will be much more poorly assessable. Very unpredictable effect on retention, again, is a sense that the trial is over. A quality of evidence for licensure will only be marginally different than that for the EUA because of what we're giving up. And actually, it weakens the scientific value of the trial that's pledged to the participants on enrollment. And this can easily be done if they're good reasons, but shouldn't be done if they're good alternatives, particularly ones that give them the vaccine anyway. So finally, this may make placebo-controlled trials more difficult for other vaccines. And we have a very strong interest in developing good information for those other vaccines, because there will be a precedent that as soon as something has been shown to be effective and is available, that it's unethical to ask people to wait any more time to be immunized in any way. And this is a precedent you may not want to set. I have a couple slides here on what the value of having more vaccines is. I actually think this committee knows it very, very well. They can have different immunization properties. There are, we might find that they're better in combination, and they might have different safety profiles and acceptance. They might have different distribution and uptake. But I'm not going to go through this, but you have these slides available. So I wanted, the last few slides, I just want to talk about the ethical impact of the unblinding, which is, or in the trust, in the whole trial system. Immediate unblinding and vaccination could become a precedent and a de facto expectation for others, perhaps undermining either ongoing or new placebo-controlled trials. A sense could take hold that even temporary withholding of vaccination within a trial is unethical because it was done in preceding trials. I would suggest that the images of, we have to remember that what happens, what we're doing here, if we do unblind, is we're disturbing the priority setting. That is, we are going to be vaccinating young, low-risk trial participants, and this will get out in the community, very different than the pictures we saw after the authorization last week, where it was healthcare workers and others. So the images of young, low-risk trial participants being knowingly vaccinated before much higher risk community members could adversely affect trust in the fairness of the vaccine testing system and the allocation system. We'll then start looking very closely at the trial recruitment and enrollment procedures of all trials to see how we are choosing people who are in the trials who are then will jump the queue. And that is a scrutiny we may or may not want to have done as so aggressively, that the enrollment in the trial is, in a sense, a privileged position with regard to trial vaccine administration. And it may be dangerous to have different ethical evidential trade-offs made in each trial by each company, and there are a lot more coming, as you know, thereby also bypassing societal priority setting for vaccine access. These priorities are generally regarded as fair, partly because the processes that created them is perceived as fair. If these are overridden in individual trials, there can be very unpredictable effect on perceptions by groups underrepresented in the trials and by the public. And if these trade-offs are trial and company specific,
then there'll be a rush by some current and prospective participants to game the system in their favor, because everybody will be looking for the trial that does better by them, thereby undermining an ethos that we're all in this together and that we need to act collectively for the greater good. So this trial-by-trial resetting of the ethical priorities and prioritization system, I think, is something that we have to think about from a large, with a wide lens. And here's the widest lens, which is the evolution of designs that we're going to be experiencing as EUAs are issued. And we're right now at this very, there we go, this very early point where there was no EUAs or vaccine is unavailable to some folks. And that's the only context in which we can do these placebo-controlled trials. And we're getting the benefit of those right now, but we're very rapidly, in fact today and last week, moving into this new era where there's some EUAs available, some vaccines available. And then we might find that the deferred vaccination RCTs planned from the inception, not this conversion, will become the standard. That is, we believe that we can reasonably ask people to put off vaccination for a few months, but in the consent, it will say after a few months you will be vaccinated. I believe we're going to very rapidly move into that, and that asking for these trials to be in that category would be a great precedent to allow that transition so future studies can be designed with that. Finally, once we get the BLAs issued, the approvals, we're probably going to have to move into an era, and I don't know exactly when this will be, but probably sooner rather than later, where the only RCTs we're able to do in populations that have the vaccines available are active control RCTs. And for these, we really need good surrogate endpoints. We really need good correlates of immunity, and that's what we're getting, that we have the opportunity to get right now. And if we undermine that, it's going to weaken the interpretability of the later active control RCTs. So I'd like to strongly encourage you to take the long view, look at this as a vaccine development ecosystem, and then if we can keep the same standards for all of the trials, particularly the priority setting for vaccine administration, and as I mentioned, none of the current international priority setting agreements include participation in a trial as a priority, unless, of course, you fall in traditional high-risk groups. So if we're consistent across all trials, I think they will both be more comparable, we will enhance trust in the system, and we'll be consistent across the board. If we start making it company-by-company, trial-by-trial exceptions, I think we're going to run into, rather quickly, problems with trust and retention. So with that, I will thank you for listening yet again, and I want to, in particular, thank the participants in the trial so far who've enabled us to have this conversation today, whose contribution was and will continue to be a tremendous gift to all of us. Thank you very much.
Thank you very much, Dr. Goodman. You went over a bit, but this was a very important presentation for our further thinking. I just want to reiterate what you have said, and that is that the crossover is not really just one design. It's a design which will vary by when the crossover is done. Am I correct in that?
Yes. And one thing I'll also add is that it doesn't necessarily have to be for individuals. It could be that the crossover is staged by priority group. That is, it's pre-planned. Every 2 weeks or every month, the next priority group will come in. But yes, it does depend on when it's done, and you want to maintain it as long as you can, as long as it's practical.
And this could be part of future consent forms, so you don't have the logistic challenge now of reconsenting individuals.
Yes, absolutely. This could be pre-specified, and I think as the design of the trial, that the trial from inception is designed this way, and it seems inevitable, honestly, if we're talking about converting these placebo-controlled trials
into this, it's hard to imagine doing a complete placebo control trial going forward. So I do think that that is what we will evolve to, but that's something you can discuss.
Okay, we have about 10 minutes now for discussion, but we're going to circle back and rediscuss all of this after we hear the sponsor presentation. Okay, Dr. Chatterjee, please. And I think some people haven't muted their phones.
Dr. Goodman, thank you for your presentation. I wanted to ask this question last week, actually, which is it not going to be difficult to maintain the blind in any of these crossover trial designs that you're talking about because of the difference in the adverse events? The vaccines are clearly much more reactogenic than the placebo. So at least the recipients and presumably the people conducting the trial would become aware or could guess which product they received.
That's absolutely. Oops. Sorry. Absolutely, I do think for a certain subset of folks, they will be able to guess more often. But remember, there's a fair overlap in symptoms. That is, even things like chills, which you would think would be vaccine specific, are occurring in the placebo group as well. So I don't, it is true that that symptom and others occur more often, absolutely, in the vaccine group. But the occurrence, but of any of these symptoms won't necessarily unblind, and still it won't be as complete an unblinding. It's really all relative, obviously, is that you tell people this is what you got. People may well suspect that. I don't think they necessarily act in ways that assume that they were vaccinated. For example, if that happened, you would imagine that people in the current trials or in future trials, when they got those reactions, would go out with very high-risk behavior. That's really what we're trying to avoid. But absolutely, that all I can say is it will occur more with the unblinding than it will with a continued blind. And we're trying to preserve as much as we can for valid inferences. But you're absolutely right. This is not, and it's true also in therapeutic trials as well. So it's a relative issue. It's not that it's completely unblinded with the blinding. That blinding is perfect, but it's much better. And also there's this other sense of sort of staying within the experimental framework and not leaving the trial and making it impossible. We haven't even talked about further follow-up, whatever, and retention for all the other endpoints. So you're right about that, but I still think it's preferential from from an evidential point of view than basically just telling everybody which arm they were in.
Thank you.
Dr. Meisner.
Dr. Goodman, I would like to thank you again for another very thoughtful and clear presentation on this really difficult issue. I'd like to make a comment, a question. As a former chair of the Vaccine Injury Compensation Program, it's important to have as careful an understanding as we can reasonably acquire from longer-term follow-up of vaccinees. I think the Vaccine Injury Compensation Program has had an enormously favorable impact on the uptake of vaccines in the United States and has resulted in the highest uptake of vaccines in history. So I would certainly encourage the blinded crossover design that you have proposed, because that may give us some opportunity to evaluate long-term complications between a vaccinated group and an unvaccinated group. And So I would just like to support that. And then the second question I have, and it may not be answerable, but has Pfizer made any decision as to how they're going to follow subjects based on your first presentation? Thank you. Over.
Actually, that will have to be your last question about what Pfizer will do, and obviously today it's not about that, but you could ask the same question about what the consequences will be in the decisions for Moderna as well. I don't know. That's going to be a question for the FDA. I don't know the nature of their conversations with either of these companies about the requirements for the BLA. I think that's where this will play out, not necessarily in the EUA, but in the subsequent requests for the kind of information they would like for the BLA. And I don't know what is happening as a consequence of after last week's meeting, just as I probably will not know after this week's meeting, but that is something for you to ask the FDA and ask the FDA representatives what they're working for or what they think they can ask.
Dr. Kurilla.
Thank you. Dr. Goodman, this has to be one of the most insightful ethical discussions I've been privileged to listen to, so thank you for that. The question I have concerns the ongoing trials. If we were to do a blinded crossover, I'm thinking of wondering how two populations would be handled. The first is people who actually develop COVID. What do we do with them? Are they done? Do they just fall out? And the other population, because of the unreliability of serology in this regard, that unless you catch it in the very post-acute phase, you may not necessarily be able to recognize someone. There's going to be an increasing percentage within both arms from asymptomatic infection, and they may be contributing to immunity, and that's going to complicate. I'm just wondering how you think going forward that can be handled appropriately with those populations.
Yeah, fantastic question. So there was a line in my slide of what we could learn that said for the infection, for the ability to prevent infection, which is really capturing the asymptomatic, and infectiousness, other designs may be needed. So this won't necessarily capture that well unless you did very frequent serology. And even then, as you say, the serology is not perfect. So we can capture a little bit of that, particularly if we increase the number of serologies. We almost certainly can't do that if they're not retained in a semi-randomized study. And that's all I will say. Well, I think that's something for the FDA to think about for both the observational designs going forward, but it is something that could be done better within a blinded crossover, and I think more successfully than if it's not blinded. But it is an incredibly important question. We can get partial information out of it here, better blinded, but not perfect. I think we're going to have to lean heavily on other designs as well.
Thank you.
Final question. The other people who've got their hand raised, please circle back. We're going to have more discussion later. Dr. Rubin.
Thanks. I'll echo what everyone else said, Dr. Goodman. Thank you for coming and speaking with us twice. And of course, what you're arguing about is very compelling, particularly, I think, for the adverse events. I think we will learn something about that waning immunity from observational trials, but this really does preserve the ability to look at AEs. And clearly, I think it's the way that these should have been designed. But let me ask you about the logistics of implementing it now. Right now, for the Pfizer vaccine, for Moderna, should it receive an EUA, people are already getting vaccine, and they're getting vaccine in specific groups so that high-risk groups are getting it first. And we may already be losing those people. So is it, do you think, going to be practical to implement this design at this point?
So as as I mentioned, what this does is it keeps the groups equal. So yes, you might lose preferentially, for example, all the healthcare workers at the beginning. You'll lose them from both the placebo arm and the vaccine arm in terms of the blinded part. But then you have a period of time before maybe the folks in the older risk groups. I mean, there's a sequencing. So you lose them sort of slice by slice. And whether, in terms of logistics, whether you do that on an individual basis or whether you plan it into the trial and you simply say, this strata will be crossed over 2 weeks after, at 2.5 months after after the 2 months of observation of the placebo group, the next one 3 weeks later, the next one 3 weeks later. So yes, you sequentially lose from the blinded part, not from follow-up, the highest risk patients, but you can still learn a lot from those who are left. But the cohorts do change, but they stay the same. They stay comparable in the two arms.
Thank you, Dr. Goodman, and please stick around for what will be late morning for you for the discussion later on.
Okay.
Now it's my pleasure to introduce representatives of the sponsor. We're going to hear from Dr. Tal Zaks and Jacqueline Miller from Moderna. Please.
Good morning. Can everybody hear me okay?
Yes, sir. Go ahead.
Thank you. Good morning. My name is Tal Zaks, and I'm the Chief Medical Officer at Moderna. On behalf of myself and my colleagues, I'd like to thank the committee and FDA for the opportunity to present our data today. We've carefully watched and listened to the meeting last week, and in preparing our presentation for today, we've attempted to proactively address many of the topics raised at that meeting and in the days that have followed. We've been developing our mRNA-1273 vaccine with a goal to seek global licensure for the prevention of COVID-19 disease, and are here today seeking emergency use authorization based on phase 3 safety and efficacy data. I don't need to belabor the damage this virus continues to wreak directly on our health and indirectly on our society and our way of life. Since the pandemic began, we at Moderna have moved rapidly to leverage the advantages of our mRNA platform, and we've been working closely with colleagues from the National Institutes of Health to develop our vaccine. We've done so in a very transparent manner, sharing our phase 3 clinical trial protocol as well as recruitment metrics with the public. Let me briefly explain the merits of our vaccine. mRNA-1273 is based on messenger RNA, a molecule that is fundamental to the biology of every living cell and serves as the blueprint for all protein synthesis. Our vaccine uses our body's own cells to activate the immune system. It enables these cells to make only the part of the virus that is critical for the immune system to recognize, in this case, the spike protein. Importantly, our vaccine platform has some inherent safety features. The mRNA does not self-replicate, does not enter the nucleus, and does not integrate into our DNA. The manufacturing process is cell-free. It does not use products of animal or human origin, and it does not contain preservatives or adjuvants, thus avoiding some of the potential concerns of older vaccine technologies. Now, mRNA-1273 is not our first infectious disease vaccine. In fact, we've been in early phase clinical trials for the past 5 years, conducting 12 clinical trials that have enrolled over 1,700 healthy volunteers. SARS-CoV-2 is the 9th virus against which our mRNA vaccines have elicited neutralizing antibodies, and we have not seen a significant safety concern in any of our trials to date. Since the company's inception, we've been investing heavily in understanding the critical quality attributes of our mRNA medicines, and we have been using these insights to continuously improve our process and manufacturing capabilities. We've leveraged this progress at the start of the pandemic to develop a product that remains potent and stable in cold chain shipping and storage conditions that are widely available in hospitals, pharmacies,
and assisted living and skilled nursing facilities. At the point of care, mRNA-1273 can be deployed in a multi-use vial with no further mixing or dilution, while remaining stable for up to 12 hours at room temperature. Our phase 3 study, which is the basis of our presentation today, was conducted in collaboration with the NIH and in accordance with clear FDA guidance. It enrolled over 30,000 participants, and we believe the results support emergency use authorization. mRNA-1273 efficacy clearly exceeds the recommendations for an EUA and eventual licensure. The vaccine efficacy rate for symptomatic COVID-19 infection was 94.1%, with a 95% confidence interval lower bound of 89.3%. These results are clinically meaningful and highly statistically significant. The efficacy observed is broadly consistent across all evaluated subgroups. Importantly, we also observed a dramatic reduction in severe cases. All of the 30 severe cases observed at the time of primary analysis occurred in people given placebo. A reduction in total symptomatic cases predicts a reduction in cases leading to hospitalization, intensive care, and death. Finally, data from 9 weeks of median exposure in more than 15,000 people vaccinated with mRNA-1273 have well characterized the short-term safety profile. We see generally good tolerability. Most solicited injection site reactions and systemic adverse events were reported as mild to moderate and resolved quickly. It is important to note and to educate people that we see an increased rate and severity of expected systemic symptoms like headache and myalgia after the second dose. We view these as consistent with a potent activation of a specific immune response. They are transient and self-limited, and we do not see a significant safety risk. These results support acceptable benefit risk for broad population vaccination to help prevent COVID-19 infections. We acknowledge the need for longer-term safety and effectiveness data. We will continue to transparently share our data, and the independent DSMB will continue to monitor safety as well as monitoring the duration of immunity and effectiveness. And we will continue to leverage the phase 3 trial even as we amend it to enable access to participants who received placebo. Now, in this regard, we face some unique circumstances. First, as it relates to vaccine supplies, none of our trial participants would be quote-unquote jumping the line ahead of others because we have clinical trial supplies available that in fact would expire and go to waste if we don't use them. Second, all of our participants are at increased risk of infection, and many have risk factors for severe disease. One of the participants on our placebo arm died from COVID-19 during this trial. He was a 54-year-old male whose sole risk factor was diabetes. I'll defer to Dr. Baden to describe our proposed next steps on the trial, which will continue to be overseen by the DSMB and should provide significant additional data on both safety and effectiveness. Now, beyond the phase 3 trial, Moderna will conduct additional studies in active pharmacovigilance to gain a more comprehensive understanding of the vaccine risk profile over time. We are initiating pediatric clinical trials, collaborating with the National Cancer Institute to evaluate vaccine safety and immunogenicity in people with cancer, and will continue to collaborate with FDA and other agencies to gather additional long-term safety data. Here now is the agenda for the rest of our presentation. Let me now turn it over to Dr. Melissa Moore.
Hello, good morning. My name is Melissa Moore, and I am the Chief Scientific Officer of Platform Research at Moderna. I'm also a professor in the RNA Therapeutics Institute at the University of Massachusetts Medical School. Over the next few minutes, I will walk you through a description of Moderna's vaccine platform and specifically our COVID-19 vaccine, mRNA-1273. As the basis of our vaccine, we created a messenger RNA, or mRNA, that only contains the instructions to make the SARS-CoV-2 spike protein in a pre-fusion conformation. We manufacture this mRNA in large quantities in a cell-free process that utilizes no ingredients of human or animal origin. We then formulate this mRNA with lipids to form lipid nanoparticles, or LNPs. As can be seen in the electron micrograph at the bottom right, our GMP manufacturing process yields a highly consistent product, about 100 nanometers in diameter. In addition to the mRNA and lipids, the only other ingredients in the vial are water, sucrose, and 2 FDA-approved pharmaceutical buffers. Importantly, our vaccine contains no preservatives, no antibiotics, no adjuvants, and all components are rapidly cleared from the body. When our vaccine is injected intramuscularly, it is primarily taken up in the draining lymph nodes by specialized immune cells known as antigen-presenting cells, or APCs. Once inside the antigen-presenting cell, RNA instructs the cell's protein synthesis machinery to make the spike protein, which is then displayed on the cell surface. In the lymph node, this allows B cells and T cells to interact with the spike protein and develop an adaptive immune response. This adaptive immune response includes production of antibodies and the development of T cell responses against the spike protein, resulting in both humoral and cell-mediated immune memory. Once the mRNA has done its job, it is degraded. Importantly, our mRNA vaccine has no capacity to alter DNA. First, our externally delivered mRNA constitutes only a tiny fraction of all mRNA molecules in the cell. Second, our mRNA is transient and remains in the cytoplasm until eliminated by the natural mRNA decay process. To alter DNA, our mRNA would have to both gain access to the nucleus and be reverse transcribed. Our mRNA contains no signals for nuclear access and no known signals for reverse transcription. So, in summary, mRNA 1273 directly educates the immune system by instructing antigen-presenting cells to synthesize the SARS-CoV-2 spike protein. In this way, it efficiently drives an adaptive immune response by protein expression in situ. Finally, our mRNA can neither interact nor can it integrate into DNA. Thank you. I'll now pause and hand the presentation over to Dr. Jacqueline Miller to discuss mRNA 1273 efficacy.
Morning. My name is Dr. Jacqueline Miller, and I am the Senior Vice President and Therapeutic Area Head of Infectious Diseases at Moderna. I'm pleased to share with you today some of the details of our clinical development program and our key immunogenicity and efficacy results. Before moving to our clinical program, I would like to review our key non-clinical results. We generated an extensive non-clinical data package in 3 different animal models, including non-human primates, or NHPs. Our data demonstrate that mRNA 1273 induces humoral and cellular immunity, including memory B cells in vaccinated animals. We also challenged these animals with SARS-CoV-2 virus and found that the vaccine could fully protect animals at subtherapeutic doses. No evidence of vaccine-associated enhanced disease. We have recently completed our developmental and reproductive toxicology study, indicated no safety concerns. Development of mRNA 1273 has been accelerated given the COVID-19 pandemic. Nonetheless, a full development program, including phase 1, 2, and 3 studies, has been executed. Study 101 was our phase 1 dose-ranging safety and immunogenicity study conducted across 3 age strata, 18 to 55, 56 to 70, and over 71 years of age. Study 201 was a phase 2 safety and immunogenicity study. The primary focus of our presentation will be the phase 3 COVID-19 efficacy and safety study, or 301, as it enrolled over 30,000 participants, approximately 15,000 of whom received mRNA 1273. Study 301 generated the vast majority of safety and all of the efficacy. So let's begin with Study 101.
This slide summarizes the neutralizing antibodies induced by 100 micrograms of mRNA 1273 across 3 age strata. Shaded area represents a panel of titers, a range of titers from a panel of convalescent sera taken from individuals recovering from COVID-19 disease. Served as the clinical benchmark to compare immunogenicity between the doses and the samples were 23 to 54 days after diagnosis. Neutralizing antibodies were induced in all participants by day 36, or 1 week, dose 2. GMTs were comparable across the 3 age strata, including participants in the older age strata, and persisted until day 119. Now let's discuss the T-cell immunity evaluated in Study 10. CD4 T cells were further evaluated for Th1 and Th2 phenotypes, since T cells are thought to be associated with enhanced disease. The top panel of this slide represents the Th1 phenotype, and the bottom panel is the Th2. Th1 dominant CD4 T cells are induced by day 43 across age strata. Minimal detection of the Th2 phenotype. This analysis showed no evidence of enhancement. I'd now like to present the immunogenicity results for the 100 microgram and placebo groups in the Study 201. Dark blue bars represent the 100 microgram dose, and the gray bars represent placebo. By day 43, there was more than a 50-fold increase in geometric mean titers in the vaccine group. And in the placebo group, GMTs remained below the level of quantitation. So in summary, our phase 1 and 2 studies showed the induction of neutralizing antibody titers in all participants by 1 week following the second dose. GMTs were observed to be higher than those of a panel of convalescent sera, and neutralizing antibodies persisted for 3 months after the second dose across all 3 age strata. Th1 dominant CD4 T cell response was also observed across age strata, was consistent with our findings in animal models. So now, let's look at the efficacy data from Study 301. Study 301 was designed to evaluate the efficacy, safety, and immunogenicity of mRNA 1273 compared to placebo in adults at least 18 years of age who were at risk for COVID-19. 30,420 participants were randomized 1-to-1 to receive 2 doses, vaccine or placebo. Participants received the first dose on day 1 and the second dose 1 month later on day 29. Participants have been monitored for efficacy, immunogenicity, and safety endpoints throughout the study. Immunogenicity endpoints include the measure of binding and neutralizing antibodies at the indicated time points. These immunogenicity samples will also be used to assess for asymptomatic seroconversion to non-vaccine antigens. These data were not available for the emergency use submission and will not be discussed for today. Efficacy surveillance occurred throughout the study. Once diagnosed with COVID-19, participants underwent daily telemedicine visits to ensure close medical follow-up. Participants were also given pulse oximeters to measure their oxygen saturation daily. Study 301 primary efficacy objective was based on COVID-19 cases that occurred in SARS-CoV-2 seronegative participants. To demonstrate success, the lower limit of the 95% confidence interval for vaccine efficacy had to be greater than 30%. Secondary endpoints for vaccine efficacy include the evaluation of efficacy against severe disease and death, COVID-19 using the CDC case definition, and COVID-19 cases occurring after the first dose. There was also a secondary objective to evaluate asymptomatic SARS-CoV-2 infection, but the results are not yet available. Please let me review the case definition for COVID-19, severe COVID-19 disease. Primary efficacy endpoint was symptomatic adjudicated COVID-19 disease that occurred at least 14 days after dose 2. To be considered a case of COVID-19, study participant had to have experienced at least 2 systemic symptoms or at least 1 respiratory sign or symptom or clinical or radiographical evidence of pneumonia and confirmed SARS-CoV-2 infection from at least 1 nasopharyngeal. Study 301 also analyzed efficacy against severe COVID-19. Severe cases had to meet all criteria for the primary endpoint and have at least one of the 4 following criteria: severe systemic illness or respiratory failure, acute respiratory distress syndrome or evidence of shock, or significant acute organ dysfunction or admission to an ICU or death. To ensure adequate safety monitoring and to enable the interim efficacy analyses this study has been monitored by a Data and Safety Monitoring Board, or DSMB. DSMB was chartered and convened by the National Institutes of Health and is completely independent from the company. In addition, an independent efficacy endpoint adjudication committee was assembled to determine if the case definitions for COVID-19 and severe COVID-19 were met. The committee has adjudicated all cases for the primary efficacy endpoint and continues to adjudicate cases as they accrue and will ultimately adjudicate all COVID-19 cases reported. 30,420 participants were randomized in Study 301, including 15,010 subjects to each group. The full analysis set includes 15,181 participants who received at least 1 dose of mRNA-1273. The modified intent-to-treat population includes participants who had no evidence of infection prior to receiving their first dose of study vaccine or placebo. For protocol population, predefined for the primary efficacy analysis. It includes participants in the MITT who received both planned doses and had no major protocol deviations. More than 92% of participants vaccinated in both treatment groups are part of this population. Now let's return to the efficacy results. Enrollment was stratified to ensure that we studied participants most at risk for COVID-19. We pre-specified that at least 25% of our study population would include participants over 65 years of age or subjects between 18 and 65 with comorbid medical conditions. We were successful and enrolled a total of 42% of the study population in these 2 categories. Let's review the study demography by gender and age. Approximately equal proportions of males and females participated, and the mean age was 51, the range of 18 to 95 years. 25% of the study population was over 65 years of age, and half of those individuals were over 70. Age and gender distribution were well balanced. This trial included approximately 10% African Americans, 5% Asian Americans, and 21% of participants who identified as being Hispanic. This is a breakdown of the comorbid conditions reported in the study. 23% of participants overall reported at least 1 pre-existing condition. This included 9% with diabetes mellitus, 7% with severe obesity, 5% each with significant cardiac disease or chronic lung disease. A specific inclusion criterion was that participants had to be at increased risk for COVID-19. Overall, 25% of our study participants are healthcare providers, and a substantial proportion of the remaining subjects meet the definition for essential workers. Taken together, the participants depicted on this table represent more than 50% of our study population. So here are the numbers of COVID-19 cases contributing to the primary endpoint by demographic subgroups. 33 cases occurred in the elderly, including 10 of the severe cases. 42 cases occurred in people from communities of color that have been disproportionately impacted by COVID-19. This slide displays the primary efficacy results for the pre-specified interim analysis. Primary efficacy hypothesis was met. Vaccine efficacy after the second dose was 94.5%, the lower limit of 86.5%. Difference between groups was statistically significant, the p-value less than 0.0001. The incidence rate in the vaccine group was 1.8 as compared to 33.4 thousand person-years placebo group. This interim analysis was submitted as part of Moderna's EUA application, currently under review by the EUA. A second analysis was performed when the full pre-specified cohort of 151 cases of COVID-19 had accrued and the 2-month median follow-up time point had passed. This analysis was predefined in the protocol as the primary efficacy analysis. There were 196 cases, 11 of which occurred in the vaccine group and 185 occurred in the placebo group. Vaccine efficacy was 94.1%, the lower limit of 89.3%. Difference between the groups was also statistically. Incidence rate was 3.3 in the vaccine group compared to 56.5 in the. Now, I would like to show you a forest plot of various subgroup analyses we performed on the primary endpoint, stratifying the population by age, gender, race, and risk factors. All subgroup analyses were consistent with the primary analysis, lending confidence to the generalizability of the efficacy. Also evaluated the efficacy of mRNA-1273 against severe COVID-19 as a secondary objective. 30 severe cases have been adjudicated at the time of the primary efficacy analysis, and all occurred in the placebo group, resulting in a point estimate of vaccine efficacy of 100%. There was also a single death due to COVID-19 reported in the placebo group. We have also evaluated efficacy according to the CDC case definition, which required only one clinical symptom from an expanded list and a swab positive for SARS-CoV-2 virus. Point estimate of efficacy with this definition, 95.1%, highly consistent with the primary efficacy hypothesis. We have also investigated the efficacy against cases of COVID-19 occurred 14 days after dose 1 as a secondary objective. There were 11 cases in the vaccine group compared to 225 cases in the placebo group for an overall estimate of vaccine efficacy 95.2%. The result is limited by the fact that not all cases are adjudicated. More than 96% of participants received their second dose, and the analysis included cases which occurred after the second dose. Nonetheless, the fact that the efficacy estimate is so consistent with the primary analysis is Kaplan-Meier curve. The cases that occurred in the modified intent to treat cohort since randomization are shown on this slide, supporting the secondary efficacy analysis for efficacy after the first vaccination. Based on this, we also evaluated the percentage of subjects in the modified intent to treat cohort according to the CDC case definition which occurred after randomization. We'll see on the next slide. So these are all the cases reported in each group, stratified by 2-week intervals up to the second dose. Overall, prior to 14 days post-dose 2, there were 62 cases in the placebo group as compared to 8 cases in the vaccine group. Most of the cases in the vaccine group were reported in the first 2 weeks after vaccination. Taken together, these analyses suggest that protection may begin prior to dose 2, but for maximum protection, both doses should be given. Our protocol-specified analysis on the efficacy against asymptomatic infection was not available at the time of the EUA submission. However, did collect pre-dose 1 and pre-dose 2 swabs for SARS-CoV-2 virus and have performed a descriptive summary comparing the number of positive swabs as a way to estimate asymptomatic infection. Among baseline negative participants, 14 in the vaccine group and 38 in the placebo group had evidence of SARS-CoV-2 infection at the second dose without reporting symptoms. There were nearly 2/3 fewer positive swabs in the vaccine group as compared to the placebo group at the pre-dose 2 time point, suggesting the possibility for prevention of asymptomatic infection. So in conclusion, mRNA-1273 has demonstrated clear and compelling evidence of vaccine efficacy against symptomatic COVID-19 disease. Vaccine efficacy was 94.1%, the lower limit of the 95% confidence interval of 89.3%, successfully meeting the primary efficacy hypothesis and exceeding the FDA guidance for COVID-19 vaccines. At the time of the data cutoff, 30 cases of severe COVID-19 had occurred in the placebo group and no cases had occurred in the mRNA-1273 group. Efficacy against severe disease is reassuring about the lack of enhanced disease, and participants in this trial will continue to be followed for breakthrough disease. All key secondary, sensitivity, and subgroup analyses were consistent with primary analysis, underscoring the performance of the vaccine across high-risk populations. Given this high and consistent efficacy, mRNA-1273 offers the potential to address the public health crisis of COVID-19. Thank you. I'd like to invite Dr. David Martin, the head of pharmacovigilance at Moderna, to discuss the safety data.
Good morning. My name is David Martin, and I'm the Vice President of Pharmacovigilance at Moderna. I will review our safety results from Study 301, since that study represents 97% of total mRNA-1273 vaccine exposures. I will present the 9-week median exposure follow-up data using the same November 25th data cutoff as the primary efficacy analysis. This provides 6,579 person-years of safety data. It represents 20% more follow-up time than previously available in our EUA submission, which was based on a 7-week median. Let's take a look at the Study 301 safety data. More than 30,000 participants were enrolled and received at least 1 dose. In both groups, compliance with getting a second dose was high. About 97% of participants received the second dose. As of the data cutoff, more than 60% had completed 2 months follow-up. Now moving to the data, beginning with solicited adverse reactions captured for the entire population. Overall, there were more solicited reactions reported in the mRNA-1273 group than in placebo, with a consistently higher occurrence after the second injection. Here are the data for solicited local adverse reactions after the first injection. As you can see, the most commonly reported was pain. 87% of participants in the mRNA-1273 group aged 18 to under 65, and 19% of the same age range in the placebo group experienced pain. In participants 65 and older, 74% of the mRNA-1273 group and 13% of the placebo group had pain. Similar patterns, but much lower rates, were seen for erythema, swelling, and axillary swelling or tenderness. Overall, these reactions were mostly mild to moderate in severity.
represented by the dark green shading, grade 1, and the lighter green shading, grade 2. Grade 3 reactions, shown here in orange, occurred at lower rates. There were no grade 4 events reported. Overall, solicited local reactions were short-lived, with a median duration of 1 to 3 days. A similar pattern was seen for solicited local adverse reactions after the second injection, and again, the most commonly reported was pain. A higher percentage of participants in the mRNA-1273 groups experienced these symptoms, with an increase after the second injection compared to the first. Again, grade 3 reactions occurred at low rates, and no grade 4 events were reported. Here we're looking at solicited systemic adverse reactions after the first injection. Fatigue, headache, myalgia, and arthralgia were the most commonly reported, and they were mostly mild to moderate. Grade 3 reactions occurred at a low rate, and grade 4 were even lower. The grade 4 reactions aren't visible because they were reported in 0.1% or less in both groups. These reactions were also short-lived, lasting a median of 1 to 2 days. Here are the data for solicited systemic adverse reactions after the second injection. As you can see, there is an increase in grade 3 reactions after the second injection in the mRNA-1273 groups. Again, the grade 4 reactions occurred at very low rates. Overall, most reactions were still mild to moderate and resolved within 1 to 2 days. I'll now review the unsolicited adverse events. Unsolicited adverse events reported in the overall stage of the trial were comparable between groups. 6 deaths occurred in the mRNA-1273 group, and there were 7 deaths in the placebo group. This figure depicts medically attended adverse events by system organ class. These two were comparable between groups, and the rates were low. Here we see serious adverse events by system organ class. These were comparable and infrequent, with no terms reported in more than 0.25% of participants. Deaths were balanced between groups and were assessed by investigators as not related mRNA-1273. This slide shows solicited adverse reaction rates after any dose by baseline SARS-CoV-2 status subgroup. Rates are shown for local adverse reactions on the left and systemic adverse reactions on the right. These data indicate that individuals who were positive at baseline for SARS-CoV-2 did not experience higher rates of solicited adverse reactions than baseline seronegatives. We have actively scrutinized our safety data to identify and analyze possible cases of anaphylaxis. We found no cases suggestive of anaphylaxis to mRNA-1273. It's important to note that participants with a history of anaphylaxis, urticaria, or other significant hypersensitivity were not excluded from Study 301. There were 2 anaphylactic reactions reported as unsolicited adverse events, one in placebo and one in the mRNA-1273 arm. The placebo event occurred 10 days after the first dose. It was attributed to co-administration of radiocontrast dye, and the participant received the second dose of placebo. The mRNA-1273 event occurred 63 days after the second dose in a person with a history of asthma and allergy to shellfish. We also ran the anaphylaxis standardized measure query and reviewed events that occurred within 48 hours of vaccination. None met Brighton collaboration anaphylaxis case definition criteria. Of course, we will continue to actively monitor for these events. I'll now review our safety monitoring activities for the post-authorization period. Moderna worked hard to develop an integrated vaccine monitoring system that complements U.S. government and other established programs and is focused on identifying safety signals as rapidly as possible. This system has 3 goals. 1, to monitor for adverse events of special interest and other concerns associated with vaccines in general. We will of course look for AESI patterns in VAERS, but we will also actively monitor AESI in real-world healthcare data, as I'll explain in a moment. With respect to safety in the event of vaccine exposure during pregnancy, a developmental and reproductive study was completed in December 2020 with no adverse findings. Given the limited human exposure to date in the phase 3 trial, we will establish a pregnancy registry that includes a cohort recruited from the general population. Our second broad goal is to monitor long-term vaccine effectiveness through a study in an integrated healthcare delivery system. Third, we will identify and assess unanticipated safety signals as rapidly as possible, again by monitoring adverse event reports from the U.S. and from other countries. But in addition, using real-world healthcare data, we can add any unanticipated safety signals to the vaccine monitoring system, as I will describe. Given the recent events in the United Kingdom, we know that an active surveillance system using a large data source is critical to capture rare adverse events. We will identify expected rates of AESIs prior to vaccination using a cohort of 45 million adults from a large linked healthcare claims data source. In this scaled visual, you can see how the sample, with women in red on the left and men in blue on the right, closely matches the U.S. population. This cohort complements, but does not duplicate, the large electronic health data surveillance systems operated by the FDA and the CDC.